Hello, sunshines. Welcome back to our next 3 chapters on genetics. So the first one that we're going to look at is chapter 19 on chromosomes. I already recorded the first 15 minutes of this lecture and then realized that I didn't have a microphone selected, so therefore there was no sound. I'm a little bitter, but I'll try not to be too bitter while we talk. Okay, so this chapter we're going to look at cell division. That's going to be our big thing here. So we'll look at the shape of DNA and its importance with cell division for mitosis, that regular body cell division, and meiosis or meiosis when we're making our sex cells, egg and sperm. Okay. So first, mitosis. So this occurs in our regular body cells and results in identical body cells. So specifically, we'll have, I don't want yellow, 2 exactly identical DNA-wise, identical cells, and those are going to be diploid or 2N. They have 2 copies of each chromosome, one from mom, one from dad. So here in this picture, fertilization of the egg and sperm results in a zygote, 2N, copies of chromosomes from mom, copies from dad, and that's going to divide and grow through mitosis. So mitosis is that regular body cell division where we're growing, where we're repairing, right? And meiosis is where we're making our sex cells, our gametes, in our gonads, right? Review back to reproductive. So ovaries and testes are the gonads. The gametes they make are egg and sperm. And the meiosis process is 2 divisions, where at the end the result is 4 non-identical haploid cells. Non-identical, that's important. Okay. They are a little bit different between males and females. Females will make only 1 egg out of those 4 cells. The other 3 are just basically DNA only, and they get expelled off and broken down. They're polar bodies. And then you got 1 egg or ovum. Whereas sperm, you get 4. That's a 4. Sperm. Great. We'll look at each of these more in depth. Okay, so first we should talk about DNA, and it's actually got different forms based on what the cell is doing. So the chromosomes are these condensed, thickened versions of the DNA. It actually has a different name when it's long like this and being read. In this case, it's chromatin or chromatin. It's string-like. It's being read. So the enzymes like DNA or RNA polymerase can come in, it can attach, it can make RNA, it can make proteins. Right? So that's when the cell is actively functioning. But think about it being string-like. It's not very condensed, it's very spread out. So if we want to divide our cell, it'll be hard to do that. So what happens is we'll wrap the DNA up like string into a ball of yarn, and we'll wrap it around these proteins called histone proteins. And so those histone proteins, other cat, they've both already been here, thicken the DNA, it condenses into chromosomes. Well, chromosomes, condensed, form of DNA for cell division. Or division. And so there, it's much more organized, it's easier to send to our new cells. And when you look at cells under the microscope, you can see the DNA when it's in chromosome form much better. And so now all along the gene, or the chromosome, I gave away what we're going to talk about, are areas that code for different proteins. Each of those areas is a gene. Let's do It's a specific segment of DNA. It codes for a specific protein. And we'll look at this more in another chapter, but basically we'll take that gene, we'll read it, we'll make a copy of RNA, we'll edit that, and then we'll make protein. So a karyotype is when we've taken all of our chromosomes and we've lined them up. And how we number chromosomes is by length. So the first pair is the longest one, and then the 23rd we'll talk about on the next slide is our sex chromosomes. So you can see in a regular average human we've got 23 pairs of chromosomes, right? Lovely. So our somatic regular body cells have those 23 pairs, 46 chromosomes, and we've got 2 copies of each, right? One from mom, one from dad. And so they're the same chromosomes, they have the same genes on there, but they could be different versions. So what we call those are homologs or homologous chromosome pairs. So 2 copies of chromosome 1 is an example. Homo same, so they're the same chromosome. You're not very comfy, are you? I'm sorry. Okay. So we've got 23 pairs, homologous pairs. Okay. And so 22 of those pairs are our regular functioning body cells. Chromosomes, those are the autosomes. And then that 23rd pair is the sex chromosomes, which indicate genetic sex. So XY male, XX female. There are a couple of interesting non-disjunctions there, though. The kitty wants to play, doesn't he? You should go play with him. Okay, you're both distracting me either way. Great. So now let's start to look at the cell cycle. So those somatic body cells, everything but egg and sperm, go through a cell cycle. The longest portion, why can't I write, is interphase. So that's when the cell is functioning regularly. Right? It's functioning, it's growing, it's preparing to divide. And then the cell division component is when it's actually dividing. And then we'll make 2 identical cells. That original cell can be considered the parent cell. The 2 divided cells could be the daughter cells. Okay, so interphase is not a phase of mitosis. It's that regular cell division. Okay. And so we'll usually look at mitosis either in a whitefish blastula, like a growing zygote has divided multiple times, and so it's an area of rapid division and cell growth, so you're more likely to see cells in phases of mitosis in those. But this cell here, here's the nucleus, right? We can see chromosomes in there, but we can't really see anything going on. Well, we can't see chromosomes. We can see chromatin, right? We can see that DNA spread out. We can't see chromosomes. So it's functioning regularly. And there are 3 phases there, G1, S, G2, GAP1, synthesis, GAP2. So think about growth. Synthesis, you make a second set of DNA. And then GAP2, growth again, preparing to divide. So this is a pretty view from our book showing that chromatin, chromatin, where the DNA is uncondensed, it's string-like, it's being read. And then we divide and make a second copy of each chromosome so that we can give one to each new cell. And so our DNA is going to start condensing. It's going to start wrapping around those histone proteins and become visible chromosomes at the beginning of mitosis. There are two parts to that cell division, the mitosis, which is basically the division of the DNA, and cytokinesis, the division of everything else. Right? Separates the cells, gets the organelles like endoplasmic reticulum and things divided. Okay, so I swear this cat wasn't here 2 seconds ago. Now we're going to look at the steps of mitosis. And the steps are PMAT. P-M-A-T. Prophase, metaphase, anaphase, telophase. So if you're ever trying to think about the order, PMAT. Okay. So in prophase, that's the first part of mitosis. Our DNA is condensing, it's thickening into chromosomes. So here's our nucleus, and you can see now there are little black squiggles. Those are the chromosomes. That nuclear envelope, the membrane around the nucleus, is also dividing. It's splitting up. Nuclear envelope, the membrane around the nucleus, is dissolving. And then we're going to have these centrioles, the centrosomes, start to go to each end. And those are going to be what organize the DNA, and you'll see why that's important. Okay, wow. I just can't. Okay, so centrioles. Centrosomes, centrioles, centrosomes. Opposite ends of the cell. This cat is annoyed with me for not holding him the way he wants to be held. Cats, I love them so much. So those chromosomes go to the opposite ends, and they're going to start sending out what are called spindle fibers. So these microtubules are going to organize how the DNA divides. Okay. So next, those spindle fibers are going to grab those copies of the chromosomes. And remember, there's two identical copies of chromosomes attached together, and they line up at the middle for metaphase. Ooh. So prophase, PMAT is first, a pro is the best, they're at the top. Metaphase, they line up in the middle. Okay. Okay. And so you've got the two copies of the chromosomes. They're going to be attached in the middle, the centromere, to the spindle fibers. So PMAT, PM, the next is A, that's anaphase. So anaphase, I like to think of, we've got two A's dividing by an N, right? So the N is splitting up the A's. So here we're splitting up the identical copies of the chromosomes. So you're going to have one copy of each going to each new cell. Identical copies